All right. Welcome, everyone. Very excited to be here today. I'm Michael Graham, and I have the pleasure, along with Dr. Scott Hamilton and Amanda Kahn, to introduce today's defense speaker. Unlike our illustrious Dr. Hamilton, I do not have all the bells and whistles, but I do have the same etiquette requirements that I must read to all of you. All audience participants will be muted throughout Acy's presentation today. Please do not attempt to turn on your video or share your screen during his talk. There will be an opportunity for you guys to ask questions at the end. Once Acy has finished his talk completely, you can do the Zoom raise hand feature, and I will then call on you, ask you to unmute and ask Acy his question, and then he'll go ahead and answer your question. And then you're invited, once we do get to that question session, for everyone to go ahead and turn on their videos. Also want to just say, when this is all done, we're going to boot everyone off the Zoom except Acy, myself, Amanda, and Scott, so that we can have the mandatory committee meeting afterwards. So we're very excited for today, and we look forward to all of your questions when we're all done. So, Acy. I don't have a whole bunch of slides to show up for Acy like Scott does, so I just want you all to simply stare at Acy. Just kidding. Or his title, it doesn't really matter what you're staring at. But Acy is probably one of our greatest success stories. And I want to talk to you a little bit about how he got here and what he's done. He came to us from Western Washington University, where he graduated in 2016 with a biology degree and a marine emphasis. He did spend some time before coming here with the Seattle Aquarium as an instructor and camp counselor. And then once he got to Moss Landing in 2018, key year there, 2018. So it's 2020. It's been 4 years, including COVID, which is absolutely fantastic. He spent some time at Monterey Bay Seaweeds, our local seaweed farm, helping us out. He was also a volunteer coordinator for the Monterey Bay National Marine Sanctuary Exploration Center, which he worked for 3 years out there, which was great. I know it was very important for him while he was working there. In addition to that, he won a prestigious CERC fellowship. CERC is the Smithsonian Environmental Research Center, and he was a fellow out there for 2020 and 2021, again, key years. And since that time, and after all this culmination we have here with his graduation and his final certification, he is now an aquarium biologist at the Vancouver Aquarium. So we're very excited that he's already into the workforce. And again, you know, Acy's just an ultimate success story. I gotta tell you a couple of stories here. The first one is, and this is really about adaptability and perseverance. Acy shows up as a beer pig at a party at my house that my wife Erica and I are throwing, and our parties are kind of fun. And Acy showed up early. It's the first time he's been there, and he walks in and says, "Hey boss, what can I do for you?" He always calls me, "Hey boss, all right, here you go, Acy." I handed him a spoon and like 100 abalone, and his job was to shuck abalone for a gut content analysis, where we would throw the abalone onto the barbecue, and then they would all go into our guts. And Acy was like, sweet. We showed him how to do it, and he just went and rolled, and it was a fantastic party. And I just thought, this kid's great, right? He takes guidance, and then he runs with it. And so think about this now. He's got his CERC fellowship. He's already got a proposal ready and running on the plasticity of Fucus, a seaweed that would be recruiting to these artificial Olympia oyster reefs that the CERC program was putting out in San Francisco. And they were rolling and going, and then boom, March 16, 2020, COVID happens. Not only were there logistic issues with his project that were just driven by the nuances of doing field work, but CERC was shut down, we were shut down, the whole planet was shut down, and Acy was in the middle of everything. So what does he do? He pivots to what I am going to just very affectionately call a backyard thesis. He calls me, well, he basically texts me a series of panic texts going, boss, what the hell am I going to do? And he's like, I'm in the middle of this. There's no way we're getting out of this anytime soon. And he came up really fast with a plan. We got him a scope. He gets his own incubators, which I think are now in someone's house holding Pinot Noir at the exact right temperature.
Mhm.
And he managed to pull off lab work at home, plus a ton of field work. And he as well as his colleague Sloan, were in the same boat. They were the first guinea pigs really to get through the COVID RISCA approval process to be able to do this stuff. And, you know, he does all this work with the support of his committee. And here we have him today. And he's going to give a fantastic talk, whether it was COVID or not. And I just hope everyone appreciates what this kid has done and how proud we are of him for his thesis. So Acy, you're awesome. I'm really excited to see your talk on the effects of habitat and temperature on reproductive success of Fucus distichus in Central California. Go get him.
Wow. Well, thank you so much for those kind words. It's easy to forget all the stuff that those years involved. But thank you so much. With that, we'll go ahead and get started. Hello and welcome. My name is Acy Wood, and today I thank you for joining me as I present my findings for my master's study, the effect of habitat and temperature on the reproductive success of Fucus distichus of Central California. I want to take a second to welcome my distant guests. It wouldn't be me if I don't throw in at least one word joke in there. As Mike mentioned, I joined the Biology lab back in 2018, and since then I have been studying Fucus distichus. It's gotten to the point where I slowly branched into almost every part of my life, and today I'm excited to share my findings with you and hopefully have you become one with the Fucus, just like how it has for me. Before we get started, I would first like to go over the agenda for the presentation. First, I'll share with you the background to my research and what motivated my study. From there, I'll review the research objectives I work towards. Third, I'll go over the method I used to address those objectives, followed by a summary of the results that method produced. Finally, I'll discuss the meaning of those results and conclude with the implications of those findings for the field. If at any point you feel lost, that's totally okay. Please look up to the top left for a reminder of which section we're on. With that out of the way, let's begin. For the last century, the world has experienced climate-related increases in environmental temperature, intensification of storms, and proliferation of heating events. These climatic trends are triggering significant responses from Earth's environments. The range and composition of multiple ecosystems are dramatically changing as they acclimate to new baselines. Impacts to oceanic conditions are causing significant shifts in global marine biodiversity and to marine ecosystems. Environments like the poles and deep sea, which both act as heat sinks for the planet, are experiencing rapid changes as they try to handle an increase in atmospheric CO2, temperature, and shifts in salinity. Ecosystems found at the world extremes are not the only ones at risk, though. Coastal ecosystems in particular are experiencing dramatic change due to the nature of their conditions. Changes in temperature, sea level, and salinity threaten to redefine coastlines and consequentially the organisms that are found there. The inhabitants of the intertidal are commonly existing at some sort of temperature tolerance threshold, and even a slight shift in an environmental condition can be a game changer. Climate change can be the cause of that shift. For example, a warming planet is predicted to affect salinity throughout the oceans. As conditions warm up, evaporation will increase. Depending on the location, this can affect local salinity in a few ways. If evaporation exceeds precipitation, the already saline conditions will become more saline through this process. The opposite is also a risk. If precipitation exceeds evaporation, then local conditions will get fresher. Phenomena like this are a threat to any organisms that rely on a certain local salinity to persist. In particular, the impact of climate change on key intertidal organisms, such as foundation species found along coastlines, are of concern. If key community members can no longer persist in the environment due to shifting conditions, its dependent community members will also experience detrimental effects. Due to being important agents within their ecosystems, any effects of climate change on coastal foundation species will trickle down and be felt by the entire community. Let's define what a foundation species is a little more, though. Foundation species are organisms that are integral for both the generation and identity of an ecosystem. Their existence creates the ecosystem they belong to, and without them, their dependent ecosystem wouldn't exist. This concept was first identified in deep water benthic environments and is now recognized to occur in ecosystems worldwide. Foundation species characterize and modify their environment by creating physical structures and modulating ecosystem processes. For example, in Figure A, corals in a coral reef build and maintain coral reefs. Mangrove trees in Figure B do the same, along with kelps in kelp forests in Figure C. Due to their nature as the framework of an ecosystem, any change in a foundation species physiology or range will reverberate throughout the ecosystems that they build. For example, let's consider a forest. Forests are defined as an area dominated by trees. Without trees, you do not have a forest. You may have grasslands or bushlands or some other ecosystem that's similar, but without the trees, the forest doesn't exist. They are central to the identity of the ecosystem. Aside from providing sustenance for multiple community members, the trees also serve a variety of roles that allow the forest to be what it is. For example, the forest floor is shaded by the canopy, cooling it down. The branches provide structures to allow animals to construct homes, such as bird nests, or aid them in securing food, like spiders building webs to catch prey. The roots can alter the soils to provide necessary conditions for other flora. Trees are critical to the forest biome. If removed, the complex web of interactions within a forest ecosystem would collapse. The shading effect is lost, eliminating any benefit to community members that needed it. The structures for nests disappear, forcing animals to relocate to other areas. Any nutrient fixing will cease, eliminating a vital source for plants that relied on it. Any change in the environment that makes it inhospitable to trees would consequentially be felt by all other community members within the ecosystem. Now that we've taken some time to unpack the term, let's review what qualifies a foundation species. Foundation species can be defined by 3 criteria set by Ellison 2019. First, they must be numerically abundant and account for the majority of the biomass within the ecosystem. For instance, in this picture of a coral reef, the area is dominated by corals. Similar to the tree example, the corals are literally creating the ecosystem from their sheer biomass. If added together, the corals account for the majority of organisms found in this photo. Second, foundation species are typically at or near the base of directional interaction networks that characterize ecosystems. This means foundation species are either primary producers or are found in lower tiers of the food network that the ecosystem they spawn produces, such as oysters in this picture of an oyster reef. While oysters are not primary producers, they still are relatively low on the food chain and can serve as a food source for other organisms. Third, their relationships to other species in an ecological network mostly reflect non-trophic or mutualistic interactions. This means that foundation species are doing more for their ecosystems than just being a meal. Foundation species are serving multiple roles within their community and don't just provide food for other community members. Usually, they're engineering their environment in some ways, such as slowing currents, seen in the kelp in this kelp forest, providing shade, or fixing nearby nutrients. Many seaweeds, such as kelps, meet the criteria for foundation species and create complex ecosystems, such as kelp forests. Another family of brown algae, fucoids, are also excellent foundation species and are great species of interest to measure the impact of climate change on coastal ecosystems. Members of this family occupy niches ranging from the subtidal to the upper intertidal, are typically abundant when present, enjoy a large foundation of previous literature regarding life history, and are easy to instigate propagation in controlled settings. Fucoids enjoyed a large, well-established library of work that has investigated the numerous roles that fucoids serve within their ecosystems. For example, fucoids create 3D heterogeneous community structures that diversify the environments they grow in. Fucoids inhabiting the intertidal have been found to provide shade and shelter to organisms that would otherwise be exposed to desiccation and predators. This combination of engineering ecosystems, supporting literature, and accessibility makes them ideal candidates to explore the effects of climate change on coastal ecosystems. Numerous intertidal and subtidal species have been studied for this purpose around the world. I would like to touch briefly on the fucoid life cycle before moving on. The fucoid life cycle is relatively simple compared to other families of algae. The production of gametes occurs in pits called conceptacles, flask-like cavities that house the production of antheridia and ogonia. Conceptacles can either be dioecious, with separate conceptacles developing either antheridia or ogonia, or monoecious, developing both in the same conceptacle. Typically, conceptacles are found on the branching tips of fucoids, usually located on swollen appendages called receptacles. Seen on the left side of the diagram, the almost Mickey Mouse-shaped tips, circled in red, are the receptacles. Mature Antheridia and Ogonia are shed as conceptacles swell with mucilage when receptacles are exposed to low energy water environments, such as during low tides or within pools of water. Shedding during periods of minimal water movement reduces gamete dilution and increases successful fertilization. Once shed, the Antheridia and Ogonia burst, releasing spermatozoids and eggs into the water. Fertilized embryos will settle onto nearby surfaces and develop into germlings, then mature adults. Typically, individuals will take 2 to 3 years to sexually mature. The cycle will then repeat. The strategy of synchronizing fertilization with events of low water movement allows established populations of fucoids to reliably recruit new generations of fucoids to the local area over time. If fucoids are unable to complete this cycle, such as failing to reliably recruit due to shifting conditions, their dependent ecosystems will gradually lose their foundation and eventually disappear. This cycle is actually expected to be impacted by climate change. Shifting global conditions are actually predicted to allow some fucoid populations to expand their range, while others are expected to shrink. Intertidal fucoids, especially upper intertidal ones, are adept at managing heat-related stress. They can withstand extended periods of heat exposure and desiccation, then return to functioning as normal when resubmerged in seawater. As coastlines warm, fucoids are predicted to experience a poleward expansion as new coastline becomes suitable for their survival. However, as with many organisms, there is a limit to what fucoids can withstand. Populations that occupy southern ranges are predicted to be adversely affected by summer heat in particular. It is simply too hot, and individuals are unable to recover and maintain photosynthetic activity. Previous literature investigating impacts on fucoidian life history have explored how adults will fare, and is a wonderful base to start to answer how coastline ecosystems will respond to climate change. But I ask, how well will fucoids reproduce as conditions shift? Adults may be able to survive estimated conditions, but will they be able to continue the reproductive cycle? To address this inquiry, I turned to San Francisco Bay. San Francisco Bay is home to a well-studied fucoid, Fucus distichus. Coastlines throughout San Francisco Bay rely on Fucus distichus to engineer and maintain a viable environment for the residents of the upper intertidal. Fucus distichus serves critical roles such as providing shelter from desiccation, protection from predators, and lowering wave energy for their environments. Local Fucus distichus populations occur in a variety of environmental conditions, such as occupying brackish water to full marine conditions, or low wave energy to high wave energy environments. San Francisco Bay Fucus distichus also exhibit low genetic diversity. For the most part, they are genetically similar throughout San Francisco Bay. This means that any differences in life history can mostly be attributed to environmental conditions. These factors make San Francisco Bay Fucus distichus an ideal candidate to investigate how coastal foundation species will be impacted by climate change. To begin to better understand this, I asked myself, how do temperature and habitat type affect reproductive success of Fucus distichus in San Francisco Bay throughout the recruitment season? To answer this question, I laid out 3 research objectives. One sec. Okay. My first objective was to examine differences in reproductive output of Fucus distichus within and between 3 coastal habitat types located throughout Central California. This objective was created to investigate if habitat type accounted for any possible differences in reproductive success of Fucus distichus, and also to begin to determine any possible effects of being from the same genetic population, but raised in differing salinities. My second objective was to examine the influence of temperature on the reproductive output of Fucus distichus of Central California. The purpose of this objective was to observe if the increase in global temperature will have any significant effect on the reproductive success of Fucus distichus. A clearer understanding of how the Fucus distichus life cycle will persist will be elucidated by comparing the reproductive output of Fucus distichus in conditions that resemble current ambient to predicted changed conditions. Finally, my last objective was to explore the interaction between environment and temperature on the reproductive output of Fucus distichus of Central California. Realistically, environment and temperature are tied to each other. Habitats do not always stay at the same temperature whenever you visit them, and a given temperature does not indicate any particular habitat type. Exploring any possible relationship between these 2 factors is vital to properly identifying important effects of either single factor on Fucus distichus reproductive output. These objectives were investigated by using a fairly straightforward method. A 4-step procedure was used to gather data. I'll review and elaborate on the steps one at a time. The first step was to identify and gather samples from the field. I began by searching the Central Californian coastline for populations of Fucus distichus. Over the course of a month, I searched every beach, bay, cove, and cranny I could find from Marin County all the way down to Santa Cruz. It took a very, very long time. I also inspected the shoreline of San Francisco Bay from Palo Alto up to San Rafael Bay. Throughout this journey, I cataloged locations with beds of Fucus distichus large enough for consideration. Sites then had to meet certain criteria. I had to be able to access the Fucus distichus population within a relatively short time upon arrival to ensure all sites could be visited in a single tidal cycle. An abundant population of Fucus distichus had to be present, and I had to be able to return to the lab before samples began to expire or release gametes. Eventually, I selected 9 sites total spread across 3 different habitat types: outer coast, inner coast full salinity, and inner coast low salinity. I'm going to describe each of these one at a time. Outer coast sites were locations along the open ocean. All have established Fucus distichus populations located throughout the upper intertidal that are exposed to full marine conditions along the outer coast. My 3 outer coast sites were Davenport, Whalers Cove Beach, and Pigeon Point Bluff. Second, my inner coast full salinity sites were located at or near the mouth of San Francisco Bay. All have bands of Fucus distichus in the upper intertidal. Previous studies have determined the salinity of these sites within this area to have similar or the same salinity to the outer coast sites, meaning that they were full-blown marine. The 3 sites were Point San Bruno, Pier 94 Wetlands, and the Degassing Station. Last, the inner coast low salinity sites were located within San Francisco Bay, but were in more estuarine conditions and have sparse populations of Fucus distichus throughout the upper intertidal. The 3 sites were Coyote Point, John and Jean Starkweather Beach, and San Quentin Beach. The order of sites was chosen to allow a single vehicle to be able to depart Moss Landing with a single person, sample all sites, and return to Moss within a single tidal cycle. I began travel between sites right as the tide began to drop, usually between 2 to 5 a.m. Starting from Moss Landing, I traveled to Davenport, then to Whalers Cove Beach and Pigeon Point Bluff sites. Next, I crossed over to the mountains and visited Coyote Point. Traveling up the coast from there, I then visited Point San Bruno, followed by Pier 94 Wetlands. Although seemingly close to each other, I would then spend at least an hour traveling through San Francisco to the Degassing Station. Sorry, there we go. Once done there, I would cross over the Golden Gate Bridge and head to San Quentin Beach, and lastly, John and Jean Starkweather Beach. The last 2 sites occupy the same purple circle. Traffic was such that heading across the Bay to Richmond, then driving back to Moss was the fastest way home. So I essentially was doing a large loop terminating at the labs. Sample collection involved running a 10-meter transect parallel to the shoreline through the bed of Fucus distichus at a site. 10 points along the transect were randomly generated in advance. At each point, a 0.5 squared quadrat was placed. All individual Fucus distichus were counted within the quadrat, with an individual being defined as a holdfast and possessing 4 or more receptacles. Each individual was given a number, and then a random number was generated to determine which individual would be collected. Keeping in line with other studies, individuals were collected and placed in a bag for a total of 10 bags per site. All bags from a single site were then wrapped in a seawater-soaked hand towel, then stored with a container labeled with the site name. Site containers were placed in a cooler for transportation. These methods are standard and have been used for similar studies for the last 60 years at least. I referred to a round of collection, culturing, and monitoring as a cycle. A cycle was conducted roughly every 2 weeks from June 2021 to September 2021 over the course of my study. I was able to complete 7 cycles of data. 2 cycles occurred in late spring, early summer, 3 occurred during midsummer, and 2 occurred during late summer, early fall. This brings us to our second step. The second step was prepping samples for culture. This was done by cleaning them and setting them to release gametes. After coming back to the lab, samples were washed by hand under cold, running fresh water to remove debris, invertebrates, and other algae. This event also capitalized on Fucus distichus's life history. A rapid shift in salinity causes the membranes containing developed gametes to burst, releasing spermatozoids and unfertilized ova into the environment. Usually, this is associated with a low tide event exposing fertile adults to the air or isolating them in tide pools, alongside a large deposition of fresh water from rain or nearby waterways, leading to a drop in local salinity and consequentially the release of those gametes. For this experiment, this process was replicated by washing fertile receptacles with fresh water, then letting them dry out to instigate release of gametes, as is standard practice in fucoid studies. Samples from all sites were kept separate from each other. 4 receptacles were then selected from each individual. If an individual had more than 4 receptacles, each receptacle was given a number, then 4 were randomly selected. Receptacles were then placed in petri dishes, blotted dry, then left to dry for an additional 30 minutes. Last, 25 milliliters of sterilized seawater was added to each dish. The seawater had been sterilized and ran through a 20 picometer filtered seawater filter and was added to each dish using a glass pipette. Then each dish was covered with a petri dish cover. Since I was alone for most of 2022, I got used to having seawater containers around, such as the one that you see here, but I had to start labeling them as people started to visit again. Don't worry, the vessel has been sterilized before holding seawater, but many people have reached for the seawater container, expecting it to be something else. Next, samples were placed into incubators. The two temperature treatments were used for this study. The first was 12 degrees Celsius, which was representative of local conditions throughout San Francisco Bay. The second treatment was 15 degrees Celsius, which IPCC models predict will be the case by the end of the century, according to certain models. 12 and 15. Since 10 individuals were collected per site and there are 9 sites, a total of 90 individuals were collected per cycle. At 4 receptacles per individual, that's a total of 360 receptacles per cycle. 5 individuals per site were randomly assigned to one of the 2 temperature treatments, so both 15 degrees Celsius and 12 degrees Celsius received a total of 180 receptacles. Following protocol established by studies also utilizing fucoids, cultures were grown into haired thermoelectric incubators, which have a programmable temperature range from 10 to 19 degrees Celsius. The incubators were internally lined with aluminum to ensure exposure to lights and to prevent external light from coming in. Each incubator was equipped with LED lights set to a 16-hour light, 8-hour dark light cycle. The 5 dishes from each site were randomly stacked and placed into an incubator set to their treatment temperature. Placement of the site stacks were randomized during each cycle. The last step was observing the samples and counting the total number of eggs and fertilized embryos they output. Once seawater was added to dishes, gamete release could begin within 30 minutes, depending on the site of origin, and once instigated, could last for multiple days. Dishes were observed using an inverted microscope every 24 hours for a period of 96 hours. After the observation period concluded, dishes and samples were discarded. 90 dishes in total were observed every 24 hours, during which my phone's camera was used to take photos. I needed a photo to help separate sites within locations, so later when I was looking at them, I knew where one ended and one started. So a simple flip of the camera was used and a photo taken. Here is a sample of some of those photos taken. Yeah, kind of fun. 2 metrics were observed. The first was reproductive output, defined as the total number of unfertilized eggs and fertilized embryos per dish, counted at 400 times zoom using the inverted microscope. If you refer to the reference photos, figure A is an example of an unfertilized egg. They are round and brown. Figures B and C are examples of fertilized embryos. They either have began to polarize, seen as the almond-like shape with a clear tip in figure B, or have a clear cell division present, which the arrow in figure C is pointing to. The total number of all 3 categories comprise the reproductive output metric. Observing this total allowed me to track a discrete amount of effort that receptacles were expelling. The second metric used was fertilization success. Embryos were viewed at 1,000 times zoom to determine if polarization was occurring or if cell division was present. Fertilization success was calculated by determining the ratio of fertilized cells to reproductive output, the total number of unfertilized eggs and fertilized embryos. This ratio allowed me to contextualize how much reproductive output was actually successful in advancing to the next stage of life. The 96-hour data was used to run a series of three-way ANOVAs to investigate the effects of habitat type, temperature, cycle, and any possible interaction between those three fixed factors. Post hoc multiple comparisons and magnitude of effects tests were used to determine important treatment combinations. Woo! All right. The results of the three-way ANOVAs differed between metrics. I'm first going to go over the reproductive output three-way ANOVA. As a reminder, reproductive output was the total number of unfertilized eggs and fertilized embryos. Before I review the data, let me first orient you to the figure. The x-axis shows the outer coast as OC, inner coast full salinity as ICFS, and inner coast low salinity as ICLS. The bars above the box plots indicate significant differences, with the number of stars indicating significance. 1 star means a p-value of 1 star means the relationship had a p-value of less than 0.05. 2 means less than 0.01. 3 stars mean less than 0.001, and 4 means less than 0.0001. The main effect of habitat type on reproductive output was found to be significantly different between habitat types. All three habitat types differed from each other, with the inner coast full salinity exhibiting the most reproductive output, having on average 591 total eggs and embryos. The outer coast had the second highest, with an average of 460 eggs and embryos. And finally, the inner coast low salinity had the lowest, with an average of 177 eggs and embryos. The main effect of temperature was also found to be significantly different between treatments. The 15 degrees Celsius treatment had higher reproductive output, with an average of 461 eggs and embryos, while the 12 degree Celsius treatment had an average of 363 eggs and embryos. Lastly, the main effect of cycle was found to be significant. The blue bar crossing the plots right here, the blue bar crossing the plots represent the overall average reproductive output of all cycles. Cycle 2 was different than all others, while the remaining cycles 1 and 3 through 7 were all similar to each other. Cycle 2's reproductive output was lower than all the other cycles. While I didn't expect cycles to differ, cycle 2 nonetheless was. In the discussion, we'll talk about why cycle 2 may have been a little bit different. Cycle 1 had an average of 520 eggs and embryos. Cycle 2 had 261. Cycle 3 had 381. Cycle 4 had 422. Cycle 5 had 492. Cycle 6 had 393. And finally, cycle 7 had an average of 428 eggs and embryos. Only one two-way interaction was significant between the three factors for reproductive output. The interaction between temperature and habitat was significant, while the interactions between cycle and either temperature or habitat type were insignificant. However, when all three factors were investigated together, a significant interaction was found. This interaction was the only one for reproductive output involving cycle that was significant. This figure portrays the three-way interaction of the three factors. The x-axis is cycle number, and the y-axis represents the total number of unfertilized eggs and fertilized embryos. Dashed lines with a triangle represent 15 degrees Celsius treatments, and solid lines represent 12 degrees Celsius treatments. Finally, green lines represent inner coast full salinity, blue lines represent outer coast, and pink lines represent inner coast low salinity. For example, the top line running across the top is going to be the 15-degree treatment for inner coast full salinity, whereas this bottom line here with the solid line would be the 12 degrees treatment for inner coast low salinity. Here you can see 15 degrees Celsius lines generally stay above 12 degrees Celsius ones, and then for the most part, habitat types layer one on top of each other. We can see the inner coast full salinity sites are usually above the outer coast sites, and both are above the inner coast low salinity sites. The order of symbols are similar to each other for most cycles. However, the symbols in cycle 2 indicate that inner coast low salinity 12 degrees Celsius is below outer coast 15 degrees Celsius and almost on par with outer coast 12 degrees Celsius, while inner coast full salinity has a much higher reproductive output than all the other site temp combinations. Next, I'll go over the results of the fertilization success three-way ANOVA. To refresh our memories, fertilization success was a ratio of fertilized embryos to total number of unfertilized eggs plus fertilized embryos. The main effect of habitat type on fertilization success was significant, with outer coast and inner coast full salinity differing from inner coast low salinity. Just like with reproductive output, inner coast low salinity had the lowest fertilization success. Outer coast and inner coast full salinity did not differ from each other, though. Both outer coast and inner coast full salinity had higher fertilization success than inner coast low salinity. Outer inner coast had an average fertilization rate of 0.88. Inner coast full salinity also actually had an average fertilization rate of 0.88. And finally, inner coast low salinity had an average fertilization rate of 0.81. The main effect of temperature was also significant. Similar to reproductive output, the 50 degrees Celsius treatment had higher rates of fertilization success, with an average fertilization success rate of 0.87, while the 12 degrees Celsius treatment had a fertilization success rate of 0.84. Finally, the main effect of cycle was also significant. Cycle 2 was different than all the other cycles, similar to reproductive output. However, cycle 3 also differed from all the other cycles. Cycles 1, 4, 5, 6, and 7 were all similar to each other. Cycle 2's fertilization success rate was lower than all other cycles, while cycle 3's fertilization success rate was lower than all other cycles except for cycle 2. Cycle 1 had an average fertilization rate of 0.9. Cycle 2 had an average fertilization success rate of 0.69. Cycle 3 had a rate of 0.84. Cycle 4 had a success rate of 0.88. Cycle 5 had a fertilization success rate of 0.91. Cycle 6 had 0.89. And finally, cycle 7 had an average fertilization success rate of 89. Two-way interactions existed for fertilization success. However, they were different than the ones for reproductive output. Only interactions including cycle were significant, while the interaction between temperature and habitat type was insignificant. Finally, the three-way interaction of the three variables was again significant. This is a visualization of the three-way interaction for fertilization success. Once again, cycle is on the bottom, while the y-axis is fertilization success. The patterns seen from the ANOVA are visible. The outer coast and inner coast full salinity tend to occur near each other per temperature per cycle, while the inner coast low salinity usually is below the other two habitat types within a temperature treatment. Also visible again is the clustering of data in a cycle that is different from the others, this time particularly seen in cycle 3. So what does this all mean? Great question. First, let's review the goals of the study. The goals of this study aim to better elucidate how critical ecosystem builders are currently set to handle shifting environmental conditions by observing differences in reproductive output of the foundation species Fucus distichus set in current and predicted climate conditions. As a foundation species, changes in reproductive output of Fucus distichus could affect the resulting ecosystem. Fucus distichus is an excellent example of a coastal foundation species that is poised to increase its geographic range as global temperatures rise. Although new areas of coastline may be forecast to become suitable habitat for Fucus distichus, no work had been done to evaluate if Fucus distichus would be able to successfully reproduce in predicted conditions and spread to predicted new shoreline areas. My first research objective addressed the effect of differences in habitat type on reproductive output and fertilization success. Both three-way ANOVAs indicated that both metrics changed depending on habitat type. More saline conditions outperformed fresher conditions, which is particularly interesting in the context of Fucus distichus's life history. Large depositions of freshwater are important events that aid in gamete release. A drop in salinity causes the cell membranes of developed gametes to burst, releasing spermatozoids and oogonia into the environment. Usually, this is associated with a low tide event, exposing fertile adults to the air or isolating them in tide pools, alongside a large deposition of freshwater from rain or nearby waterways, leading to a drop in local salinity and consequently the release of gametes. Because of this, I'm not surprised that the habitat types with more marine conditions have higher reproductive output. That change in salinity is going to be stronger than habitat types where individuals are in more fresh conditions typically. In a warming climate, the atmosphere can hold more water, leading to expectations of more evaporation in regions where evaporation is dominant over precipitation, and more precipitation where precipitation exceeds evaporation. In the ocean, this translates to salty gets saltier and fresh gets fresher. This is to say, based off of these findings, in the future, as saline habitats trend towards becoming more saline and fresher conditions trend towards becoming fresher, according to my analyses, Fucus distichus reproductive success will either increase or decrease accordingly. The second research objective addressed the effect of temperature on reproductive output and fertilization success. Both my analyses indicated that the two metrics will increase with elevated temperature. This aligns with the current state of the field, which predicts the fucoids are pre-adapted to warmer climates and will fare well as conditions warm. As an upper intertidal alga, Fucus distichus has adaptations that allow it to handle heat-related stress for prolonged periods of time, which is a valuable asset in a world that is heating up. Part of the reason behind the increase in reproductive success is again linked to the gamete release mechanisms. The membranes that keep mature gametes bound break easier in warmer conditions, which results in higher temperature environments, exhibiting increased rates of overall output as those bonds break and fertilization as more gametes are released. For the most part, cycles were similar to each other. Cycle 2 was significantly different than all the other cycles within both ANOVAs, while cycle 3 was different than all the others only for fertilization success. The way cycle interacted with other variables differed between the two metrics. It only factored into the throughway interaction for reproductive output. However, for fertilization success, it was involved in every interaction that was significant. Future works could incorporate more cycles to clarify if cycle 2 was simply an outlier or if a larger effect was in play. My last research objective addressed the interaction of habitat type and temperature on reproductive output and fertilization success. Both metrics were found to have significant throughway interactions, but seemingly for different reasons. Let's begin to explore the interactions in more detail by looking at reproductive outputs. A relationship was found between temperature and habitat type, meaning that reproductive success at a given habitat type will change depending on the temperature at which it is being measured, and vice versa. No other significant two-way interactions existed. Notably, a further significant interaction was found between cycle and the temperature and habitat type interaction, meaning that the interaction between temperature and habitat type on reproductive output varies depending on the cycle. Even then, it was only cycle 2's effect on the temperature habitat type interaction differing from all other cycles. The remaining 6 cycles and their effects on the temperature and habitat interaction were all similar to each other. No significant interaction was found between cycle and either temperature or habitat type, meaning that cycle only plays a role in determining reproductive output when interacting with the temperature and habitat type interaction. The middle of summer is the least optimal time for reproductive release of fucoids. So it is possible that the populations do not have mature gametes ready for release. This trend is similar to other studies and supports the findings of my analyses. It is also possible that the day was too hot for the adults. Looking at the data, the individuals who were gathered first during collection, when it was still nighttime, fared better than individuals who were collected at the end of the sample collection. The day of cycle 2 was particularly hot during summer, exactly the conditions that previous work suggest could damage the physiology of adults beyond what could be repaired in a single day. This is a potential bias in my study towards sites that are collected first and could be controlled better in future studies with a larger collection team collecting simultaneously. Unfortunately, this was not possible due to the ongoing pandemic. Cycle served a more prominent role in the analysis for fertilization success. The two-way interactions ended up being the opposite of reproductive outputs. Only relationships with cycle were significant. The lack of a relationship between temperature and habitat type implies that regardless of temperature, a habitat type will exhibit a given fertilization rate, as well as the opposite. Regardless of habitat type, a temperature will exhibit a given fertilization rate. Any significant relationship between temperature and habitat type was only found within the three-way interaction, telling me that relationships with cycle were more important within the three-way. A similar observation was seen where the comparisons of three-way interactions resulted with cycle 2 differing from all other cycles. However, unlike reproductive output, cycle 3 also differed from all other cycles. Overall, the 15°C treatment had the highest fertilization success, although which habitat type had the highest rate changed from cycle to cycle. This could be indicative that different habitat types develop gametes at different rates and have different optimal times in the year for fertilization success. The San Francisco Bay populations of Fucus distichus that comprise the intercoast full salinity and intercoast low salinity are great examples of observing the differences in reproductive output and fertilization success. All the Fucus distichus populations of San Francisco are almost genetically identical, meaning that any differences observed between populations are due to phenotypically plastic responses, probably influenced by the environment. The intercoast full salinity populations had better overall reproductive output and fertilization success than their intercoast low salinity population counterparts. But populations from both habitat types had greater reproductive output and fertilization success in warmer conditions. Regardless of habitat type, reproductive success should improve as temperature increases. The pattern of more reproductive success under warmer temperature coincides with previous studies investigating the effect of climate change on fucoid population and range. Any shift in population dynamics of a foundation species will ripple throughout the resulting ecosystems that they engineer. The creation of new populations or increase in population size creates more suitable habitat for the ecosystem the foundation species builds. The predicted increase in range and reproductive success should result in similar expansion opportunities for organisms that reside in Fucus distichus beds, or lead to novel interactions as analogs of existing community members take residence in the habitat that is created. To generate accurate estimations of what future Fucus distichus beds may look like, the life histories of existing members should be investigated through similar climate-driven lens, determining if predicted shorelines are suitable for physiological processes will continue to operate as they do. Depending on how existing community members respond to climate change, new Fucus distichus beds may have different community compositions and dynamics. Overall, my research objectives were explored, and I was able to reach conclusions. Research or reproductive success of Fucus distichus within Central California is affected by the environmental conditions of temperature, habitat type, cycle, and their interactions. All had significant influence on the reproductive output and fertilization success of Fucus distichus. Receptacles exposed to warmer temperatures had greater reproductive output and fertilization success. Receptacles from more saline habitat types outperformed receptacles gathered from estuarine habitats. Lastly, an interaction between temperature and habitat type exists for reproductive success, but not for fertilization rate. The results of this study indicate that San Francisco Bay Fucus distichus should flourish under predicted climate change regimes. Fucus distichus is poised to experience increased rates of reproductive success as the potential range of the species increases over time. The conclusions from my study align with similar literature addressing fucoid life history that predicts a successful expansion of range. Fucus distichus of Central California will be able to successfully reproduce in predicted environmental regimes, especially as temperature increases, which should allow them to persist in any novel ranges they transition to. My findings also find that more saline conditions outperformed fresher conditions. And as models predict that saltier places will get saltier, while fresher places get fresher, it's possible that depending on local conditions, Fucus distichus populations will experience an increase or decrease in reproductive success. Fucoids have been defined as pre-adapted to deal with climate change, and this study adds to the growing body of research that affirms those hypotheses. There is a lot of potential to continue this line of research in other studies. My study was constrained to the coastline of Central California, but Fucus distichus can be found throughout the Northern Hemisphere. The same study could be repeated throughout Fucus distichus's range to observe if similar findings are found throughout the range or if they are specific to Central California. Other studies investigating fucoid responses to climate change found that different populations of the same fucoid species can exhibit drastically different responses to changes in environmental conditions. Southern range populations in particular are vulnerable to changes in heat. My study focused on Fucus distichus located in the center of Fucus distichus's Pacific range. Repeating this experiment with Fucus distichus closer to the equator, the southern range of Fucus distichus would be very interesting. It is also possible that warmer conditions have a cost that couldn't be detected within the 96-hour window that I observed my embryos at. It would be very interesting to continue to observe Fucus distichus past the zygotic stage to determine if they successfully develop to adulthood or if some sort of impediment develops, barring them from completing their life history. Lastly, this study can be repeated with multiple species of fucoid to determine if the observed effects can be generalized to the whole family or if they are specifically found within Fucus distichus. Even within the Northern Hemisphere, a wide range of fucoids can be found, and it would be really exciting to see if they behave similarly. With that, I would like to thank everyone for joining me today and to take a few minutes to acknowledge and thank a few people. First off, I'd like to thank the Moss Landing Marine Labs community. Coming here has allowed me to make so many new connections and friends, from my D&D pirate gang friends to COVID roommates to randomly made friends in the hallway. I met so many cool people. Each class walked through the halls, and seminar was an opportunity to meet someone new and look at a new perspective. All the staff were wonderful and helped me whenever I needed it. Special shout out especially to Tara for always having a solution to every problem I ever had. I would also like to thank Mike and the Beer Pigs. Y'all taught me how to engage in critical thinking on the fly, and it is a very valuable skill. Everyone always had such good ideas and helped me raise my project out of the ashes over and over and over again. Mike warns us that beer pigs will invade your life, and I couldn't ask for a better group to do that. I would also like to thank the Smithsonian Environmental Research Center's Invasive Species Lab. Dr. Zaven gave me my huge break that eventually blossomed into this study. Working with them allowed me to learn all about San Francisco Bay and find out where I could find shockingly vibrant beds of Fucus distichus throughout the area. The stars aligned to allow me to follow them into their lap, and I couldn't be more grateful. Next, I'd like to thank all the non-Moss people who helped me through the hardships. From old friends to new ones that I'm making here up in the Great White North, you all have helped me recover from hard days and share the pains with me for many a drive looking for beaches. I would also like to thank my parents who never stopped believing in me. We all, everyone says they're not going to do it. We all do it, dude. From mom calling after collections, going, "Hi, honey," to dad calling up saying, "Hey, son." Even though they were usually far away, you always felt nearby. Y'all were always happy to come to my little seaside shack and chill. Thank you so much. I'd also like to thank the rest of my family for their support, letting me crash whenever I needed it all throughout California, or giving me an excuse to go get fried food because they were in town. Y'all made me feel really loved, and I appreciate you so much. Last, I'd like to thank my wonderful partner, Kyle. I could not have accomplished this without you. You helped me get back up every time I felt like giving up. From COVID delays to remaking entire projects to getting lost in the coding. Thank you for making me emergency bolognese so I had something to eat after my 18-hour days, and setting up my Target pickups and giving me recipes for what I just got because I was too tired to do anything, let alone find food for myself. You always inspired me with how hard you work, and your standards for every step of the scientific way always allow me to just raise what I'm doing to the next level. I love you, and thank you so much. With that, I'm done. Does anybody have any questions about anything?
All right. Excellent. Thank you, Acy. I am going to allow everyone to flip on their videos if they would like, and start raising hands if they do have any questions. There are applause coming through, so I'm going to go ahead and do mine loud, Acy.
Thank you so much. I appreciate it.
Well done.
I can see my parents driving. Hands on the wheel, Dad. You're driving.
Yeah, exactly right. As we wait for other questions to come up, I'm going to start. As you know, I've always got this interesting kind of view on how beer pig science will relate to restoration and conservation implications. And your position at CERC was always an interesting one because it was inherently part of that conservation and restoration component. And in the end, it didn't ultimately work out, right? The whole oyster reef project and et cetera. But yeah, when you look at what you've done and you looked at your various habitats, that is, open coast and intercoast and full salinity and low salinity, I look at all three of those as possible places that people may look for restoration activities, right? For example, the oyster reefs that would be going in some of those low salinity and full salinity intercoasts. And so given that, and let's say you and Kyle make it back to California, or maybe you make it somewhere up there that has Fucus in a job where someone's going to ask you to help them design modules, right? Say those oyster modules you were doing, where they specifically want to encourage Fucus. Mm-hmm. Give us an example of how you could spin your research to give different restoration mechanisms, whether you're doing it in the full salinity or in the high salinity, based on what you know about this critter. So again, let me just trying to restore in high salinity, trying to restore in low salinity, oyster reefs, but you want Fucus to come in. How do you go about doing that based on what you now know?
Sure. Yeah, I would say you're probably going to have your most success from transplanting adults. People have been having a hard time transplanting germlings from a lab setting to wherever they're trying to restore. And if you're going to be transplanting adults, you're going to want to be transplanting multiple at a time. They work together to help break up wave energy. So regardless of if you're in a high or low salinity setting, if you don't have enough individuals to protect against incoming wave energy, then things are going to get obliterated relatively quickly. I would also advocate for really looking into what kinds of surfaces you're going to be using and at what angles. Typically speaking, you don't see Fucus at much more of an angle than just a gentle slope. Hard surfaces or hard vertical surfaces are really hard for them to persist on. And so I would say you're going to want to design a system where you're not experiencing too much high wave energy at first to allow those transplants to actually survive the transplantation process. It's a little stressful. Then yeah, you're going to want to make sure that you're setting yourself up, you're set up for success by having sediments at angle that actually supports future recruitment. When taking into consideration for high versus low salinity, I would say that you're going to want to consider nearby inputs of fresh water. You're also going to want to consider what your environment is going to look like at low tide. You want to make sure that your adults are able to get dried off, and they're also going to be experiencing any rain or any possible way for fresh water to be coming in. If they're not drying out all the way, a little bit of mucilage can still come out, but it's not as efficient as if they're drying out, then getting that freshwater submergence. For low salinity is going to be a little bit tricky. As my results indicate, you're still going to have some output and fertilization success. It's just not going to be as high. By nature of how the gametes are developing within the conceptacle, if you don't have an intense enough change in salinity, then you're not going to get a very large output of gametes into the environment. And that can dilute what you are actually experiencing immediately outside of the adults. With that dilution, you're going to have a decrease in, or a potential decrease in fertilization rate. Or I'm sorry, oh yeah, you're going to also have a decrease in fertilization rate as gametes have a hard time finding each other, but you're going to have a harder time getting multiple recruits. And as these things start to senesce, you need to have that new generation coming up, so that way you actually have full coverage. That kind of ties back into what I was saying of making sure that you're preventing too much wave damage from coming in. If you're not recruiting enough, then eventually, as you lose members of the community, that wave damage is just going to come over and just take the rest out. So yeah, just keep in mind your slopes, keep in mind your freshwater inputs, and I would say those are the major things.
Right on. So before I ask your committee members if they have any questions outside of the committee meeting, I'm going to go ahead and let Dan, who's got his hand up, unmute and give you a question.
Sure.
Hey, Acy. It's people always say it's not going to happen to them, but dude, yeah, it happens to everyone. I know it. I want to talk to you about cycle 2. Sure. So was there any indicator that there could have potentially been some sort of release right before you collected that could have contributed to that much lower overall loss of reproductive output compared to the other cycles? Was there anything like abiotic or tidal or freshwater input or anything that could have signified that?
Just to clarify your question a little bit more, so you're asking if during cycle 2 any factor that the individuals experienced before I got there could have resulted in gamete release before I got there?
Pre-collection, yeah.
Let's see here. I mean, it's possible. That day was just particularly really hot, and if I remember right, the low tide coincided with the middle of the day, which is a big bummer for intertidal organisms, especially. So if I had to guess, I don't think it was so much so that gametes were already released when they came out. There wasn't a lot of freshwater input that day in the local areas. Rivers were low and hadn't rained in a while. I genuinely think things were just fried. I think the physical structures inside the conceptacles didn't fare very well. The adults were very crunchy that day. They were all starting to turn black. I think they were just scorched. And I think because of that, there was just not as much activity being released, and whatever was coming out probably wasn't competent. If you look over at the slide that I pulled up, the intercoast full salinity 15-degree slides or for that day, they still had relatively large amount of input coming in. This includes sites that their orientation was such that they were still relatively shaded when I visited them. So I don't think they were as strongly affected by the sun. This is all conjecture, though, without being able to test this further or set out objectives specifically to address that. I wouldn't be able to know for certain. There is also the possibility that something happens during my traveling around. Maybe my car got too hot and destroyed things before I got back to the lab, or there could be a lot of different explanations. I couldn't pin one specific reason as to why cycle 2 stood out as much as it did. I lost
Thank
my
you.
Zoom thing, so
Sorry, I'm still here. So Scott and Amanda, if you guys do have questions now, I don't see anyone else with questions, but if you have a questions for outside committee, feel free to let him have it. Otherwise, I'm going to ask another one to close it out. All right, Amanda. Unmute yourself.
Perfect.
There you
Hello.
go.
Hi, Acy. Great job. This was so interesting to read about and now to see you present it. And I'm super interested in thinking about foundation species as well. And you cited some of the same authors that I've read who talk about foundation species that build their foundational habitats along with other foundation species, which in the intertidal is a really big deal, right? You've got mussels, you've got barnacles, you've got fucus. So I'm curious about how you feel about the habitats that are being built by these multiple foundation species, and sort of whether you envision fucus's presence or absence or changes in its presence or absence as affecting the other foundation species and the other communities that form, kind of whether the mosaic approach or whether the nested assemblages approach or ideas might fit.
yeah, I think that's really cool. One of the benefits of Fucus is that one of the reasons why it likes to be on a gentle slope is that as the tide comes out, it will lay down. And as it lays down, it's trapping moisture underneath its fronds. And that's one of the ways that it helps its community members all around it. So I definitely could, that was one of the reasons why the Smithsonian actually were curious to see if Fucus would allow oyster reefs to have increased and recruitment and retention of oysters. So I definitely think from what we were able to see from that, which granted was limited due to transplantation, it did help with recruitment and it did help cool them down. And so in the context of other invertebrates that are sessile, that are trying to survive until that water comes back, I definitely think any Fucus that is able to help retain that moisture and help them stay cool during that time is going to have a compounding effect with each other and support each other. I don't know, I can't think off the top of my head of any mutualistic benefit from the oysters or barnacles onto Fucus, other than possibly helping to also ameliorate wave energy. That seemed to be the biggest deal for what I saw going around. If there was too much wave energy at a site, then the fucoids would experience sublethal biomass loss at first, but if it was sustained, then eventually that could lead to some serious problems. So those hard-shelled organisms being able to take those hits, I would imagine, helps them survive that environment. Does that address your question?
Yeah, it's just nice thinking about the different ways that those different foundation species might interact and whether you get any kind of synergistic or additive effects by having them change in their relative abundance to each other.
For sure. One thing that also makes me wonder about is looking at things that utilize Fucus as a shelter that then could also go eat the mussels and the barnacles, any snails or any other organisms like that that could be using the Fucus to hide from the sun or stay moist during low tide. I think that's really cool. That warrants further investigation.
Great, thanks.
Right on. All right. So I think Scott, Amanda, and I will go to committee, but I want to ask one more question. You know, aquaculture is on everyone's mind. Acy, you've had some interest in it. I've had some interest in it. You know, Scott and I have a grant where we're using aquaculture to grow bull kelp for restoration purposes. So everyone's getting excited about it, right? And people often ask me, why don't you guys farm fucoids, right? And I always say, God, they're a pain in the butt. And, you know, kelps are so much more prolific and easy. You just got to, you know, play the backside of Led Zeppelin and fertilization occurs. I mean, it's crazy. And yet what you showed is maybe that's not necessarily the case. And if fertilization success is bad, maybe in these low sites, outplanting individuals isn't going to be a good mechanism because you're going to outplant them. They're going to have low fertilization success. Can you briefly describe if for any young entrepreneurs, there is a possibility that the aquaculture industry can benefit or not fucoid restoration in these areas where you're going to have problems like San Francisco Bay?
Yeah, I could make an argument for that. For the restoration work that I did utilize in Fucus in San Francisco Bay, we did have to go identify sites that one had healthy enough individuals to transplant in the first place. And, you know, by nature of transplanting, you're taking away from that healthy area and you're bringing it to a place that hopefully it will do okay with, assuming it survives the transplantation in the first place. So just that alone incentivizes someone to possibly look into lab conditions to grow things for outplanting. The difficulty with it is getting fucoids to release their gametes is easy. That part is pretty well done. And having them survive to the germline stage is easy enough. It is at that point where their phenotypic plasticity usually kicks into their environments. So getting them to you would have to find up a way to characterize where you're transplanting to, then attempt to recreate that successfully in a controlled setting to hopefully get the plastic response you're looking for, that once you transplant it out, it's able to do what it needs to do. You can have a drastic change in size, the density of fronds, how thick the midrib is. There's so many different morphological characteristics that can be produced. And it's not a perfect science. We know that wave energy, for instance, dictates generally how big fucus will get, fucoids will get, but the other characteristics not as much. So it could be that you're raising fucoids wrong per se, and then you're throwing them out into an environment where their body plan is wrong for that area, and they may not be able to persist. So yeah, I would say it's in theory doable. You just have to put a lot of effort into recreating faithfully in the lab the kind of site that you want to bring those buddies to.
I like it. So actually, I'll take the analog for some shellfish and fish species that you can actually get fertilized eggs in the laboratory quite easily. The problem is the settlement, the morphogenesis, and then the ultimate grow up to adult size. That it sounds like what you're saying, that's probably the bottleneck for fucoids. Whereas in kelps, you just let them go and they're off and running. All right, well done. So that ends it. Anyone who is still here, if you want to undo your video and show your faces and unmute, I'm going to give us a solid 20 seconds of screaming and clapping for Acy. So just want to congratulate him one more time. Go ahead and unmute, turn up the volume, scream out if you want. Good job, Acy.
Thank you so much. Thank you. I really, really appreciate it.
Good job, Acy.
Good job, Acy.
Oh, I hear mom screaming. Mom and dad are screaming.
Good job.
Yeah, man.
Thank you. Thank you so much.
Clap,
I
clap,
really
clap,
appreciate
clap,
it.
clap.
There's more. Don't
Thank you,
crash the
everyone.
car. Bennett and Andrew are driving down the highway. All right, now we're going to kick you all out, and I'm getting